In step 7, we are ready to add the plasmid DNA to our bacteria and calcium chloride in the plus DNA tube. Teachers typically keep the plasmid DNA on ice to distribute to groups. Adding your plasmid DNA is pretty straightforward if you have access to and know how to use a micropipette. If using a loop to add your plasmid DNA, dip the circular end into the plasmid DNA provided by your teacher. You should see a film like you might get on a bubble wand. Then dip that into the tube labeled plus DNA and twist. The DNA will come off into our cloudy mixture of bacteria and calcium chloride. As you can see in step 8, you want to leave your tubes in ice for 10 minutes. Be sure not to just lay the tubes on the top. The bottoms of your tubes should be well buried into the ice. In step 9, have one person set a timer for 50 seconds. Bring your ice cup over to the 42-degree bath. Float both tubes for 50 seconds. After your group member tells you it's been 50 seconds, put both tubes immediately back into the ice for 2 minutes. It's very important that after those 2 minutes, both tubes are taken off of the ice and placed into the tube rack for step 10. In step 10, we feed the bacteria in each tube with 300 microliters of LB broth, again using either a micropipette or a transfer pipette. Mix the bacteria by turning them upside down several times. In step 11, the bacteria recover in the incubator at 37 degrees for 5 minutes. As much as possible, keep that incubator closed during this time to maintain its temperature. Unlike our previous time steps, it's okay if this is not exactly 5 minutes. Between 5 and 20 minutes is just fine, as the cells are happy and warm in the incubator. While the bacteria are recovering during the incubation in step 11, you have time to move on to labeling your plates as described in step 12. A few tips for labeling your plates in step 12. First, you'll notice that there are 2 different types of plates. If it has a black stripe on it, that means that the agar in that plate contains LB. LB has sugars and amino acids that the bacteria need in order to grow. If the plate has a black and a red stripe, it means that that plate has LB in the agar, but it also contains ampicillin, which is a type of antibiotic. When you're labeling the plates, we want to make sure that you label the bottom of the plate and not the lid. You should include this information here around the outside edge of the bottom of the plate. We don't want you to write in the middle of the plate, as that'll make it difficult for us to read our results. Once you've labeled your plates and your bacteria have had enough time to recover, you're ready to move on to step 13. Retrieve your bacteria after they're done recovering and move on to step 13, where we plate the bacteria. Begin inverting the minus DNA bacteria a few times to mix them well. We'll then use our transfer pipette to dribble 150 microliters of the bacteria onto the agar of the plates you labeled minus DNA. Remember, we want this on our agar, not the lid of the plates. We'll swirl the plates to spread the bacteria evenly. Then do the same process for the plus DNA bacteria on our plates labeled plus DNA. At the end of step 13, you will have plated 2 minus DNA and 2 plus DNA plates and are ready for step 14. Students, in step 14, you stack your plates upright with the lids on top, as we see here. Teachers, leave the plates upright as it takes about 20 minutes for the bacteria to soak into the plates. Even up to an hour is fine. Be sure to turn the plates upside down when you place them in the incubator, as condensation can drip onto your bacteria. Some tape can help keeping incubator doors from popping open if they contain a lot of plates. Our last step is to follow your teacher's instructions for cleaning up your materials, disinfecting workstations, and of course, washing your hands. We hope these tips are helpful for you and your bacterial transformation.